All right, I can go for it. Let me just start my video. I think we haven't seen. Hi, everyone. I'm Ida. Just nice to put a face on here. Today, actually, I would like to introduce you my colleague, Christoph. You heard him already. So he's our senior FAS. Hello, Christoph. And he's based in Germany. He's our most precious knowledge base in the team. And I'm very happy to introduce him, and he will be presenting how our data analysis service and our new platform, the Rosalind, can help you with your data analysis. Thanks.
Thank you, Ida, for the kind introduction. Can you see my screen?
Yes, very well.
Okay, perfect. Thanks a lot. Just a moment. Yeah, thank you for attending the NanoString Summit and for your interest to learn more about the data analysis options that NanoString is offering. As Ida said, my name is Christoph König. I'm a field application scientist with NanoString, and I'm speaking from Berlin today, and I'm happy to be here and to provide this overview to you. Just a second. Oops, I need to organize my desktop a little bit better. So, okay. So, as Michael probably already presented earlier today, NanoString is a company that focuses on accelerating the rate of research by extracting more data from less sample. Our capacity for gene expression analysis in bulk and spatial profiling is well established, and this is indicated by the great number of publications that that is using our systems. Today, we have more than 3,800 papers that were published using our Encounter platform. And we have already close to 30 publications that have appeared using our Geomics platform that was launched less than a year ago, I would say, or around a year ago. Well, next to the benefit of the technology of being able to generate high quality data from FFPE samples, another great advantage is associated to the relative simplicity of doing the data analysis. Data files are small and can be easily transferred and stored, and analysis practices are comparatively easy, straightforward, and transparent. Today, I like to focus on new options that help you to facilitate data analysis even further and support you to understand the results of your study better and faster without having to have extensive biostatistics or R scripting skills. So one important prerequisite for the uncomplicated data interpretation of Encounter data is the effort that we put into panel design. Our panels are more than just random collections of genes. Our bioinformatics team takes pride in making sure that gene selections and curation reflect latest standards and scientific knowledge. Usually, our panels are built around a scientific framework or model that is actively discussed and leveraged in the respective field, like shown here, for example, the hallmarks of cancer, a concept brought forward by Hanahan and Weinberg in 2011, or the cancer immunity cycle model proposed by Chen and Melman in 2013. Then latest publications and consultation of key opinion leaders and the respective research community are taken into account to come up with a list of genes that represent pathways and biological processes relevant for the topic of the panel in the best possible way. The assignment of genes to these pathways and processes that you get with our NanoString panels is a major facilitator to extract biological relevant information from your experiment in a straightforward way. So while looking at gene sets, at genes that act together in the same pathway or biological process of benefit for the analysis, one aspect is obviously that clear functional information is associated to the pathway assignments. The other is that looking at groups of genes that are co-regulated obviously helps to improve the statistical significance of differential expression scores. As you see in the volcano plot on the left-hand side here, for illustration, it's obvious that some of the genes show nice p-values, but if you do a multivariate testing, taking into consideration that hundreds of genes are being considered in parallel here, all candidates drop quickly well below the significant threshold here, as seen for the FDR plots on the bottom left side here. Now, when looking at gene combinations, the drop in significance during the false discovery rate correction is much less pronounced. And with strain signatures that further weigh certain genes within the group depending on their influence in the process, the scores withstand the more rigorous testing even better, as you can see here on the right-hand side for the tumor inflammation signature that holds the multivariate testing with high FDR scores at a good scale here. Now, while well-curated gene annotations that are important to identify differentially regulated molecular processes informing the analysis with appropriate sample information is of big relevance as well. Before actually starting an experiment, you want to have hypotheses that are supposed to be tested. The better defined they are, the easier it will be to draw meaningful conclusions from the results. As NanoString technology allows for relative gene expression analysis, you always want to do pairwise comparisons and check with genes or gene sets that are differentially expressed, e.g., for example, between normal and a disease state, or before or after drug treatment. Now, providing additional clinical or experimental information beyond the immediate factors believed to drive the differences between the comparison groups helps to check further for parameters that influence or even dominate the differences in addition to the hypothesized one. Principal component analysis can help here to visualize such drivers. In the plots here on the right, you see how the samples in the study are distributed in the planes of the biggest variation of the overall data in the study. There is one major driver for the variation in the data which contributes to it by 47%, as you see here with the 0.47 next to the PC1. The second largest driver is responsible for only 22% of the variation. So in the upper panel, samples are color-coded by their tumor state, so normal versus colorectal cancer here in this particular sample, while on the bottom panel, the label indicates the sample replicates or the sample origin, the patient from where the different samples actually came. It's clear to see that the samples cluster together better by the tumor diagnosis than they do by patient ID. And it's likely that the tumor diagnosis corresponds to the first principal component, causing 47% of the variation. It's, however, obvious that there's also some slight clustering by samples, as you can see here by the light purple samples, particularly here. So the patient background obviously has an influence on how the tumor tissue differs exactly from the normal one, and therefore may be considered as a confounder. Now, I appreciate that this is a rather simplified representation and explanation that I'm providing here. Why I'm showing this? My main point here is that I like to encourage you to enrich your queries that you do with the data with as many experimental and/or clinical parameters that you were able to collect. Using the software tools provided by NanoString, those may help you to understand and differentiate out further the biology or technical influences that had an impact on the results of your experiment. So what are these tools exactly? Those of you who have worked with NanoString data before are probably familiar with the standard software Insolver and the advanced analysis plugin that can be downloaded and used for free, free of charge from our website. Insolver supports you to do a basic QC, to confirm robust technical performance, and perform reference gene normalizations and basic group comparisons. Advanced analysis provides you with a rich set of graphical representations on the results of your study, like volcano plots and clustered heat maps, and it provides you with a gene set analysis or cell abundance profiling outputs. We have many on-demand video tutorials and webinars on our website that explain Insolver and advanced analysis functionalities in greater detail. We're happy to point you to those, or also to help you directly if you have questions or difficulty with using the software. We can certainly line up a web conference for that, or web call. What I like to focus on more in my talk today are two alternative options for data analysis that you may not be aware of so far. So one is the option to do our data analysis in the cloud on the Rosalind platform. We have seen in the past that sometimes installing the advanced analysis module can turn out to be a bit clunky, as our libraries have to be downloaded and placed on your computer correctly, which may not work 100% automatically with some of the configurations or network settings. Therefore, we decided to work with a professional provider for cloud data analysis to remove the need for local software installations and to leverage their state-of-the-art analysis pipelines, interface, and the knowledge base repository they're offering. You'll hear more about that in a moment. The other option that I'd like to present here today is our offer to do the data analysis for you. Dedicated scientists and biostatisticians run the analysis for you at NanoString on a fee-for-service basis. More about that in the latter part of my talk. So let's turn to the Rosalind platform first. In October, NanoString joined a collaboration with the software and data analysis company OnRamp Bioinformatics, which is based in San Diego in the US. OnRamp's mission is to enable scientists to analyze their complex genomics and transcriptomic data effortless and without having extensive bioinformatics and statistical background knowledge. And the platform was already renowned for their cloud-based analysis platform, which is called Rosalind, in honor to the co-discovery of the double helix Rosalind Franklin. Our collaboration agreement opened up the usage of the Rosalind platform to all users having encounter data, free of charge. You only need to register and to establish a user account, and you'll get access to the majority of the data analysis functionalities that this platform is offering. OnRamp Bio guarantees highest standards of data security and confidentiality, so that you shouldn't be concerned about the safety of your results and your findings. On Rosalind, you can do data QC and normalization and configure pairwise comparisons of sample groups, very similar to what is possible in Ensolver and in advanced analysis. We're working closely together with OnRamp to implement the same analysis tools, like, for example, gene set analysis or cell type profiling. So features that you may be used to from working with advanced analysis. The configuration of the jobs is highly intuitive. The analysis will take just a few minutes, and as everything is cloud-based, you don't need to worry about underlying scripts or compute settings. Exploration of the results is very interactive, and that's what you see here by this blinking screen and the blinking slide that I'm showing at the moment. Heat maps and volcano plots are provided, and by hovering over data points or rows in the heat map, you'll see which gene exactly is differentially regulated and how the p-values look like exactly. Plots, tables, and associated information can be downloaded and used for further documentation or publication. Filter criteria and comparisons can be easily changed and reconfigured to focus interactively on the most interesting aspects emerging from the data analysis process. Another nice feature of the Rosalind platform is the option not only to look at the gene annotations that we from NanoString provide with our panels, but also to reach beyond that by accessing over 50 additional knowledge bases and to check further for relevant differentially regulated biological processes. Selecting between the different databases is easy and straightforward, and genes associated to a process, as well as significant scores for the deregulation of the process, are being transparently displayed. What has been lacking in Ensolver so far was a nice and straightforward tool to further investigate the biological function of differentially expressed microRNAs. Rosalind closes this gap now with a very nice and interactive option to look at the structures, but also at the genes that are highly likely to be regulated by a microRNA of interest. So here on the right-hand side, you see a list of the mRNAs that are likely to be regulated by the microRNA that is being selected here as a target. Rosalind furthermore opens up to combine data from different experiments using different technologies like sequencing or qPCR in one meta-analysis to obtain higher level information and to integrate complex results in one joint assessment. Different from the options I showed before, this functionality, however, is not free of charge and would require an at-cost subscription to the platform. Results and data can be shared on the platform easily with collaborators worldwide. The analysis and filtering can be reviewed together and be modified interactively in a joint discussion and evaluation process. So certainly a feature that is very important and nice to have in these days of COVID-19 disease. So here's a representation of how the QC data report looks like after import of the RCC files from an Encounter platform. Tables and plots help you to review the quality and provide an intuitive source for identifying potentially problematic or outlier samples. I highly encourage you to register for the Rosalind platform and to test out its functionalities. Again, the platform is quite easy and intuitive to use, but both the NanoString as well as the on-ramp bio support are at your disposal to help you further with questions that you may have. Maybe check also on the website that I'm highlighting here on the bottom now. You find a link there to a recorded webinar from a user providing a more detailed insight into what the platform offers. And the insight is more detailed than what I can offer here in presenting the features in 10 minutes. I hear that OnRamp is also offering online webinars at the moment that are particularly dedicated to customers. So this is something you can certainly also look at on their website or on our website and enroll on participating in these tutorials, online tutorials. Sorry, that was the wrong direction. So now let's switch gears and turn away from the tools we have that enable you to analyze your Encounter data on your own. If you're not comfortable with that, or if you are dealing with a more complex analysis setup, or if you like to investigate samples using proprietary signatures, NanoString can do such analysis for you in a custom service setting. So this rather busy slide gives you an overview about the different types of projects the NanoString data analysis service is offering. On the left side, you see what we call the panel analysis service. This is an offering where we would discuss the experimental lineup and go with you and run in a default setting more or less one or several extended advanced analysis jobs and report them back to you. This panel service can be further customized so that scripts outside of advanced analysis are also being used. That, however, would come at an additional cost, obviously. In the middle here of this slide, you see our offering to run individual signature analysis for you. Here, in a research use only setting, we would run our proprietary algorithms for calculating, for example, the PAM50 score for breast cancer subtyping on Encounter data that you would provide to us from patient samples. We are offering the same for the other two clinically validated signatures that NanoString owns, the tumor inflammation signature indicating the level of tumor immune infiltration and activity in a tumor sample, and the LST test, the lymphoma subtyping test, providing cell of origin information for patients suffering from lymphomas. Our final offering, which is highlighted on the middle part on the right-hand side, is what we call our 360 analysis reports. I'll present in more detail what these reports cover in a moment, but the idea here is that we provide a comprehensive patient-by-patient as well as summarized feedback how all the signatures covered in our Immuno-Oncology 360 or Breast Cancer 360 panels behave. These reports are particularly well suited if you have data from a bigger clinical study and you like to identify biomarkers, for example, predicting survival chances under certain drug treatment regimes or indicators for drug response. With any of these offers, you receive a dedicated project consultation, you have a direct scientific point person at NanoString, and you receive back reports with publication-ready figures. So let's look closer now at what the idea behind our 360 panels and reports is and how they look like. Our 360 panels provide a 360-degree holistic view of either the tumor microenvironment and the immune response. Here are two examples for these 360 panels. On the left, we have our pan-cancer immuno-oncology focused 360 panel, and on the right, there's our breast cancer specific 360 panel. These panels have a content that is expertly curated again, covering comprehensively these areas of biology. They contain about 770 genes, respectively, across key pathways and biological processes, and in addition to that, subsets of genes are also grouped into signatures. The signatures are actually a key feature of the panels, which allow you to dissect the heterogeneous biology of a particular tumor. NanoString offers also an option for added flexibility, allowing you to spike in up to 30 custom targets to these base panels of 770 genes, covering genes of your particular interest that may not be represented in the basic panel. So when establishing the IO360 panel, NanoString has built around the tumor inflammation signature that gives an indication about how inflamed the tumor is, how well it is recognized by immune cells. It has built around this tumor inflammation signature additional signatures that allow to further qualify in detail the interactions between tumor, immune system, and stroma, and to identify possible mechanisms of immune evasion used by the tumors. We are, so to say, able to navigate through the different pathways in this diagram and to dissect and uncover the different biology and heterogeneity of a particular tumor. The IO360 panel is available for research use only, so you can profile samples from patients in clinical trials retrospectively for research, but you cannot use its information for treatment decision-making. So a similar idea and rationale was the basis for the design of the breast cancer 360 panel, although the framework and the core of this panel is a little different. Here, the foundation is our PAM50 signature, which is well known, and you may have heard that it's been commercialized as an official FDA-cleared diagnostic test named Prosigna. The PAM50 signature stratifies tumors from breast cancer patients into 4 molecular subtypes, being shown here: luminal A, luminal B, HER2-enriched, and basal-like. And depending on which subtype a patient has, there's a different likelihood for recurrence of cancer and for relapse of the cancer after surgical removal of the primary cancer. A large set of the additional signatures included in the breast cancer 360 panel now helps to further understand what the drivers are for the different subtypes you are observing, making a specific tumor cell to fall in one or the other category. They can also help to unravel biological mechanisms driving responsiveness to therapeutics in support of biomarker development. So when you run a study with clinical samples using our 360 panels, our data analysis service can effectively and straightforward read out the complex gene expression information and put it into the context of the frameworks and flowcharts that I showed in the last 2 slides. So before doing the actual analysis, a NanoString bioinformatics analyst will discuss the goal of the study together with you to specify the analysis details so that the report can be customized within certain boundaries. In our reports, we allow for 3 specific analyses that can be determined by you. These can come from different types of analysis. It can be, for example, a response analysis if you're looking at responders versus non-responders in a clinical study setting. You can do some grouping analysis if you have different biological variables, like, for example, a mutational status, and look at differences between mutated and non-mutated groups. If you have survival data, we can do a survival analysis for you and provide Kaplan-Meier curves. And if you have multiple time points per patient, we can look also at time series and do a time series analysis as well. As also highlighted on the slides here, there are different types of plots and graphs visualizing the results and helping you to understand the biology. Volcano, forest, and box plots highlighting the characteristics of differential expression between groups. We provide ROC curves, Kaplan-Meier plots, swimmer plots for survival and biomarker analysis, and spaghetti plots when looking at results from time series. So this really depends on what we agree with you on how the analysis should be customized for your specific needs. The report finally comes as an electronic file in HTML format. So it's highly interactive and you can browse between different views here. As you see on the left-hand side, there are different tabs that allow you to navigate through the data. The report starts out with an overview that provides an executive summary that's written by a NanoString scientist who reviewed your data in the context of the study goals. And it provides a summary of the key figures and the key findings in the report on this summary page. The heat map tab allows for getting a high-level overview about the full data set and to obtain first ideas of relevant biology underlying the study. So in this section, we have three types of different heat maps. One showing clusters of your samples based on the performance of all signatures. A second with the sample clustering directed by the expression of all genes individually. And the third heat map is allowing you to do a deep dive on the individual signatures, one by one, showing the expression of each gene within those respective signatures. A really nice feature that allows you to understand the heterogeneity in a particular sample is the wheel plot representation. Here you see examples for wheel plots for the IO360 report on the left-hand side, and then one for the BC360 report on the right-hand side. So these wheel plots allow you to look at unique gene expression fingerprints of each sample in your data set. If you take the example on the left, this represents one patient. In the center, we plotted the TIS score. So here the TIS score is 9.25, so it's a relatively inflamed tumor. Then around the circumference, around the axis, we plotted the signature scores for each of the signatures in the panel. And now you see which activity, which process is also more active or less active in this particular patient. And you see, for example, that certain immune checkpoint mechanisms like CTLA4 expression, arginase expression, PD-L1 expression is elevated, indicating that the tumor is obviously raising its checkpoint defenses against immune attack here in this particular patient. This allows you to do a simultaneous analysis of the scores of all signatures for a particular patient. And on the right-hand side, we have the same for the BC360. In this case, the subtype classification is taken as the center. So here we have a patient that was luminal B positive, and it's indicated here what the different levels for the other subtypes are. And again, you see in the outside here the other scores for the other signatures in the BC360 panel. So the report also comes with an extended methods section, and it also provides a summary of the statistical and analysis methods that were used. The information that can be exported directly to another report or to a publication. So again, you get publication-ready figures and graphs here. I invite you to have a look at the demo reports that can be accessed on our website. Please see the link below here that is posted on the bottom. You can download the HTML folder and then navigate through the example reports at your leisure. We are also happy to arrange a meeting with you to go over the report together and to answer specific questions that you may have around that. You can contact us, our dear colleagues at BioVitrum, to organize such a session if you are interested. Okay, with that, I'd like to conclude my talk and open the round for any questions or comment that you may have. Thanks for your attention.
Yeah, no, Alex, this is Agnese. I just wanted to say a couple of words to introduce you here, just to make sure everybody knows you. And Alex is taking care of European customers at NanoString and help them develop the projects and support them for any spatial biology needs, and has been a very important colleague for us at NanoString in helping us understand how the spatial biology is really accelerating the discovery of biomarkers and bringing to you in the field a new tool to identify and really unleash the power of spatial biology. Alex is gonna take us through a sort of an introduction. There will be something to refer to as we go along during the day and look at results that customers have obtained with the GeoMx spatial profiler. So Alex, please take it away. Thank you.
So thank you very much for everyone to attend this session today. So as Agnese nicely said, I'm going to be doing a presentation on a novel platform, which is called the GeoMx Digital Spatial Profiler. In this presentation, I will highlight the power of the system to resolve heterogeneity of tissue samples and to uncover novel biomarker signatures by taking on a highly multiplex, digitally quantitated, and morphology-driven approach to help characterizing tissue microenvironments. So in this session, I will show you how the technology itself is working, but also use some nice use cases and applications, especially in the fields of immuno-oncology and neuroscience. So as we all know, there is a current paradigm that is applied to us, from a technological standpoint, which either pushes us to do, unspatially resolved data analysis by destroying the tissue content, which is usually what you need to do when you do bulk RNA sequencing or even ex situ single cell analysis. So you get access to potentially the whole transcriptome level, but then you don't know exactly where the signal was initially coming from into the tissue sample. Or then you can take a highly spatially resolved approach, which is usually incarnated by traditional immunofluorescence microscopy, where you will have access to single cell information, but you will be extremely limited in the number of parameters that can be analyzed in one go. And this, of course, becomes extremely important to assess and work within the spatial orientation of the tissue specimen, because cells interact with one another, and we need to understand this interconnectedness between the cells in order to extract novel predictive biomarker signatures in diseases, which is highly the case in the field of cancer research, especially talking about minimal residual disease, that is a type of spatially constrained biology, or when we want to fish for specific markers, which may as well be spatially constrained by nature. So what DSP does here is to mix together the power of the nCounter platform, which is highly targeted, and work extremely well with challenging samples in which the RNA is usually pretty heavily degraded. And it combines it to classical immunofluorescence microscopy. As you can see here, by virtue of being able to acquire an image space across up to 4 different channels, we can really investigate the morphology of the sample. And then we can zero into specific regions of interest from which we want to extract a very dense layer of information. So in designing the platform, we really wanted to make sure that 3 major criteria would be met. We wanted that platform to be potentially unlimited in the breadth of analysis that it could measure, including both proteins and RNAs. Also, we wanted to make sure to extract information only in meaningful, interesting areas of the tissue specimen, as you can see here represented by the geometric regions of interest depicted on the image. And also, we wanted to ensure that the platform will be highly amenable to any type or format of samples, including formalin fixed, paraffin embedded samples, as well as fresh frozen samples. So in essence, doing DSP in the lab is pretty akin to immunohistochemistry. The only difference here, as you can see from this slide, is that we are mixing two types of regions with one another. The first type of region is called imaging regions. It's pretty standard. You have them already in the fridge. These are essentially fluorescently labeled antibodies. And as I said before, we can use up to 4 of them in order to reveal the morphology of the sample in space. And next to that, within the same cocktail, we are using the so-called profiling regions, which I'm going to be showing you in the next few slides. But in nature, by using this highly multiplexed cocktail of probes in one staining step, we can already have a slide that is fully amenable for DSP analysis. You can see the profiling regions, which are proprietary to NanoString, down here. We have 2 types of regions, depending on whether you want to measure RNAs or proteins. But the chemistry is essentially the same, as you can see from the schematics here. Key is that in place of using a traditional fluorescent molecule attached to a fish probe or to an antibody, we use a small single-stranded piece of DNA, which is synthetic by nature and is highly specific to the type of antibody or to the type of in situ hybridization probe we attach it to. And you can see as well that as part of the chemistry we use, there is a chemical linker which is highly sensitive to UV light. So as shown before, the first step involves you to stain a tissue specimen with a highly multiplex cocktail of DNA barcoded fish probes or DNA barcoded antibodies. Then we are going to be leveraging the fluorescence from the 4 channels to be revealing the morphology of the sample in space, which gives us the opportunity to select regions of interest in which we want to extract this highly multiplex information. Then the instrument will be shining UV light into precisely located regions of interest so that the DNA indexes which were formerly attached to the fish probes or the antibodies will be released and in a supernatant that sits atop of the FFP sample. And next, the instrument in an automated fashion will be collecting all of those spatially resolved DNA indexes and seclude them in a well of a 96 well plate. So we proceed in a similar manner to be now able to collect, as many hourwise as the investigator wishes to do so. And at the very end of the experiment, we're going to be using a genomics type of readout in order to be able to digitally quantify the information, meaning at the single molecule level. And for that, we can use 2 types of readouts. You're very much familiar with the nCounter readout, and that's the readout of choice when you want to do biomarker validation or biomarker research. The nCounter flexibility gives us a breadth of analysis that goes up to 96 plex, whether you measure RNAs or proteins. But if you want to do pure biomarker discovery work, meaning that you want to be fully unlimited in the breadth of analysis for RNAs and proteins, potentially going up to the whole transcriptome level, you will have to use an Illumina sequencer. And you can see that the chemistry here is slightly different. In place of using the traditional sets of hybridization probes coupled to an optical barcode for downstream digital quantification, we're going to be using the DNA index released from the tissue sample as is, and work toward building a library prep for downstream Illumina sequencing. So one of the beauties of our approach is that we are fully targeted. So we can design the entire content of panels and make sure it's fully optimized and validated for your purposes. And this is very different from what the traditional bulk RNA sequencing workflows impose onto us, where you are pretty much restricted in the breadth of analysis, depending on the availability of RNA molecules which are present in the FFP samples. And these are usually pretty heftily degraded. Here, by using a targeted approach, we ensure that we're going to be highly sensitive to any type of RNA or protein that we measure in the tissue samples, especially the weakly expressed molecules, which are typically not amenable to bulk RNA sequencing analysis. And this is nicely exemplified here, where we can easily identify and classify colorectal cancer patients along microsatellite stable versus microsatellite unstable groups by just looking at a molecule that is typically weakly expressed and not detectable by a bulk RNA-seq. So we have a full menu of applications that has been fully validated by NanoString, which currently focuses on immunology, immuno-oncology, and neuroscience. And we do that both for protein measurements and RNA measurements. You can see here in the protein lab, in the protein world, that the way we have been designing panels is highly flexible. We can pick and choose from different modules that you can see here, each containing on average 10 validated antibodies that serve different biological purposes. And by virtue of combining them with one another, we can actually reach well over 80-plex and multiplexing capability, both for immuno-oncology and neuroscience applications. And this is pretty unique. It's the only platform out there that is able to live both on the promise of being able to measure RNAs and proteins in a spatially resolved manner, which gives you the opportunity not only to do pure discovery work at the RNA level, but do direct downstream validation at the protein level without having to go through any other orthogonal method. Again, when it comes to the sample types, we are completely agnostic with DSP, which means that we can work with extremely limited amounts of sample coming off of core needle biopsies, going through tissue microarrays, and up to whole slide mounted tissue samples. And this again, regardless of the format, may it be formalin fixed, paraffin embedded, or fresh frozen. Coming now to the modalities of profiling, you can see that we have a breadth of applications represented here, ranging from simple geometric selection of regions of interest, which are going to be manually performed by the operator, going up to more complex but fully automated segmentation strategies. You can see through those segmentation strategies that we can extract information that is differentially contained in distinct biological compartments that may be tumor versus tumor microenvironment. We can also do rare cell profiling, as the resolution of the DSP instruments allows us to spot individual single cells across a wide field of view so that you can collect information that is cell type specific. We can do control profiling, which helps you determine proximal responses across a specific area of the tissue sample, or even gridded profiling, which is a deep mapping approach that generates thousands of layers of RNA information across an entire tissue image. So this is typically what you see when you do DSP analysis. We here have a colorectal cancer biopsy specimen that has been profiled for different regions of interest, having each different sizes and shapes, into which we have done further segmentation to be able to extract the information that is tumor specific in blue, or in orange, or stroma specific in blue. And then typically one of the major applications that we see emerging in the literature is to fish for novel spatially resolved biomarkers, which are predictive of therapeutic response, especially in the field of immuno-oncology. Here, typically, investigators select different regions of interest. They like to segment the information out coming off of the tumor or the stroma. And then by using highly multiplex RNA or protein profiling panels, they can get access to spatially resolved biomarker signatures, which help them to predict how patients, even before being put on treatment, are likely to respond to specific immune checkpoint inhibitors. More and more investigators are now trying to focus more and more into individual cell subset compartments. And you can see a nice example here in the field of melanoma, which has been published over a year and a half ago, where investigators from David Ream's lab have been trying to spatially resolve the information down to specific biological compartments, such as melanocytes or macrophages and even T cells, by virtue of using this combination of fluorescently labeled antibodies. Other examples exist in the literature. A nice example from the group of Jennifer Wargo at the MD Anderson Cancer Center, who is currently focusing on finding novel spatially resolved biomarkers in the context of skin cancer. And you can see here the workflow was extremely simple. Patient samples had been collected at baselines and on treatment. And by DSP, they could identify novel spatially resolved biomarkers related to B-cell biology, which is pretty new in the literature at the time. A follow-up study came through, focusing on studying the B-cell involvement into those observed positive clinical outcomes in anti-PD-1 treated patients. And as you can see here, starting from the H&E stains, where they could see a dramatic increase in the presence of tertiary lymphoid structures in the biopsy specimens, they could take on the DSP approach and finally profile within the B cells and the T cells inside the TLSs, but also outside of the TLSs, which gave them the chance actually to see how proteins were differentially expressed across the two different compartments. And what stood out is a dramatic expansion of markers associated with T cell activity, which were specifically found in the TLSs, meaning for T cells only in close proximity to the B cells. And those results were further confirmed independently by another group in Europe, driven by Goran Jansson at Lund University, where they also differentially profiled B cells and T cells within these tertiary lymphoid structures. But beyond that, they were also able to see that different types of B cells exist within the TLSs, some of which expressing high levels of Ki-67, meaning very active B cells, were actually expressing as well high levels of CD40, and therefore indicating that those B cells are potentiating tumor rejection responses by the T cells located in the TLSs. Other examples have been published since then in the field of neuroscience. You can see a very nice example here in the field of Alzheimer's disease, where the DSP technology has been used to profile within the vicinity of the amyloid plaque deposits in brain sections of AD patients. And a study here has been focusing on two types of Alzheimer's disease patients, one that is relatively standard Alzheimer's disease patients without any known mutation, and Alzheimer's disease patients featuring a variant of the TREM2 molecule, which is usually involved with microglial cell activity. And by using a contour profiling methodology, as you can see here, where you establish those donut shapes around the beta plaque deposit, DSP was able to profile in the beta plaque region itself, but also in the distant and more proximal microenvironment. And the signature established at the protein level here tell you a little bit about how differentiated those plaque regions are between the high ID patients versus the patients carrying a TREM2 variant. Many other publications have been appearing in the literature over the last few months. We have now 29 publications available on the DSP technology, both in the field of immunology, as well as neuroscience. And as you can see here, key aspect of the DSP technology is its very short turnaround time, meaning that because we have a throughput of up to 12 samples per day, we can easily go through quite large cohorts of tissue specimens and be able to publish very rapidly in high impact factor journals. Moving forward into 2020, we are now switching a little bit more gears towards the RNA readouts. RNA readouts started already last year with a panel that works well with the nCounter platform, but that is again limited to 96-plex in its profiling capability. Now, because we are using the NGS readout, we can definitely expand on the content that we built within our panels. And we started in August this year by releasing two new panels called the Cancer Transcriptome Atlas panel, as well as the Immune Response Atlas panel. I will cover details of those panels, but as you can see from the number here, both are covering for more than 1,800 genes, which are either cancer specific or immune response specific, especially in the area of COVID-19 research. And moving forward further this year, with a predicted launch early in 2021, we're going to be releasing the whole Transcriptome Atlas panel, which, as the name says, is covering for the entire protein coding transcriptome that is available both in human and in mouse. So first, with the Cancer Transcriptome Atlas, as you can see here, we have tried to pack the best of all nCounter worlds within one cancer-focused panel. The panel contains over 1,800 genes, which we measure across, contains 95 established pathways, comprising 35% of the genes that are immune-focused. We have been packing into this panel very useful signatures in the field of cancer, including the tumor inflammation signature that is a hallmark of NanoString panels, as well as the PAM50 signature that is currently being used in the field of breast cancer diagnostics as well. And as you can see, most of the IO360 panel is also present in the Cancer Transcriptome Atlas. If some genes are missing from the panel, there is space for customization. As you can see, we can add up to 60 additional genes of interest in that panel in order to be able to tailor the approach better to your particular field of interest in cancer research. And now the applications are changing a little bit because of the number of RNAs we can visualize at once in a spatially resolved manner. We're also expanding on the tools that we are that are present in the DSP software, which Aida is going to be showing to you a little bit later today. The Cancer Transcriptome Atlas helps us now resolve and answer questions which couldn't be answered before. Like, for instance, in this case, focusing on colorectal cancer specimen, we can start investigating the invasive margin in high resolution and high details, and do control profiling here, for instance, to investigate which of the most predominant signaling nodes found across the cancer reactome are differentially expressed either in the tumor area or outside of the tumor area. And of course, you can do that with all of the signaling nodes which we measure in the Cancer Transcriptome Atlas, which gives you a very nice way to very rapidly and easily spot differential expression levels for RNAs depending on the pathways you are specifically looking at. In parallel to the Cancer Transcriptome Atlas, we have been launching the COVID-19 Immune Response Atlas, which, as the name says, is a very nice panel to study the host response in the face of COVID-19 infection. This panel is not much different from the Cancer Transcriptome Atlas, but we have been adding some genes as spike-ins to be able to detect the presence of the virus in the tissue specimens, but also empowering the panel with genes associated with pulmonary activity, pulmonary immune response. And a few publications have been emerging already in the literature using that panel, which only was released over the summer. You can see a nice example here where the Immune Response Atlas panel has been applied to COVID-19 disease patients, where DSP was used to profile lung specimens in different areas of the lobe, especially in areas which were highly infected with SARS-CoV-2, highlighted here by specific detection with a FISH probe, shown here in red. And specific signatures could emerge from those areas which presented high viral burden in those selected regions of interest, potentially identifying novel biomarkers that could be leveraged in the therapeutic setting. And finally, our next stepping stone toward unbiased spatially resolved profiling is our Whole Transcriptome Atlas panel, which measures across 1,800 genes for human samples and over 21,000 genes for mouse samples. So this really makes us completely agnostic to any type of disease area or research interest. And we will have a panel launching early in 2021 for human samples, and 2 months later, one panel for mouse studies. Early data is already emerging in the literature as well as in communications. We can see a communications here made by the Sanger Institute, which stress tested the whole Transcriptome Atlas panel against other technologies emerging in the field of spatial transcriptomics. And as you can see here, they generated absolutely outstanding results by testing the technical and the biological reproducibility of the DSP approach using the whole Transcriptome Atlas panel. As you can see here, when they profile in similar biological areas of a given tissue specimen, they could generate very high technical reproducibility, which could be also visualized at the biological level by performing experiments across several sections where the very similar concordance score could be devised in otherwise identified from sharing similar morphological features. Other examples in the field of cancer using the whole Transcriptome Atlas are now emerging, where this approach is being used to understand where the pioneering cancer cells that can be found in the lymph nodes forming metastases are initially originating from into the primary tumor. By applying the whole Transcriptome Atlas and being able in one snapshot to visualize it, and seeing where mRNA spikes are precisely located across the reactome, it becomes quite easy to identify mRNA gene clusters, which are being recapitulated in different areas of the tissue samples. And in that specific case, what was newly uncovered is that the IDO1 associated mRNA genes, which are typically found in the core tumor, are also recapitulated in the lymph node metastasis, indicating that the pioneering cell forming metastasis in the lymph nodes may presumably not come from the invasive margin, but rather from the tumor center. And our next step now moving forward is to be able to leverage the existing single-cell RNA-seq signatures and put that back into the spatially resolved context of the tissue sample. And for that, we have been designing a novel algorithms called Spatial Decon, which leverages those single-cell RNA-seq signature and can help us to assess abundance scores of all the possible single-cell populations within the spatial resolution provided by DSP. You can see that here in emerging applications examples, which are available as posters right now, not fully published yet. But here we have been working together with the Garvan Institute in Melbourne towards establishing connections between the breast cancer human cell atlas and trying to find all those spatially resolved signatures back into the tissue context. You can see here that by DSP, we have been profiling across many regions of interest from which we extracted segment-specific information for the tumor and for the stromal microenvironment. And by doing this deconvolution of RNA signatures, we can now get access to the abundance of individual single cell subsets and put them back into the context of the tissue. And this, of course, becomes very powerful because then in one snapshot, you can start understanding where those single cells are located across a tissue specimen. In that specific study, this approach was used actually to identify the presence of tertiary lymphoid structures by showing colocalizations of T cells and B cells across the entire tissue specimen. So I'm reaching out the end of this presentation. These are the 5 major pillars that make the DSP platform unique in the market. It's highly sensitive by virtue of being a targeted approach, and it's completely unlimited in terms of this multiplexing capability. It does that for both protein and RNA profiling, which is unique as it gives you a tool to validate orthogonally any discovery that you may have been doing at the RNA level and extend it to the protein length. You can enrich the information across precisely located regions of interest which matter to you. you can work with any type of tissue specimen, whatever its size and embedding format, may it be FFP or fresh frozen. And finally, we deliver to you highly qualified, optimized, and validated panels so that your experiments can be focusing on the biological information right from day one without any further optimization on your end. And with that, I would like to thank you very much for your attention, and I'm happy to take any questions that you may have. Thank you. So it's my great pleasure to be introducing our next speaker of the day, Benjamin Terrier. Benjamin Terrier is a professor of internal medicine at Cochin Hospital in Paris. Benjamin specializes in management of systemic autoimmune diseases, especially systemic vasculitis, and he provides inpatient and outpatient care in the clinical department of Cochin Hospital, assisting in the management of patients through the National Reference Center for Rare and Systemic Autoimmune Diseases. Benjamin went through medical school and his internal residency in Paris from 2002 to 2008. During his time, he identified target antigens of self-antibodies directed against fibroblasts in idiopathic and systemic sclerosis-related pulmonary hypertension in the lab of Professor Luc Mouton at Cochin Institute. From 2008 on, Benjamin completed a PhD in immunology in the lab of Professor David Klatzmann in Pitié-Salpêtrière Hospital, characterizing autoreactive B cells during hepatitis C virus-related autoimmunity, among other autoimmune conditions. As vice president of the French Vasculitis Study Group, Benjamin coordinates clinical studies for patients with systemic vasculitis. Today, Benjamin will present some of his most recent work involving the use of the nCounter platform to characterize the molecular determinants of the host response to SARS-CoV-2. And with that, I'm leaving the floor to Benjamin.
Hi, everybody. I hope you hear me well and you see my slides. So thank you very much, Alexandre, for this nice introduction, and Beaucoup for the invitation. So I'm really glad to talk about the last study we performed on the COVID-19 pandemic, and especially on the role of the transcriptional analysis that was able to define the transcriptional abnormalities in this severe disease. As you know, the COVID-19 pandemic raised at the end of 2019 in China, and very quickly the natural history of the disease highlighted the fact that there was distinct patterns of disease progression. And we were able initially to identify from the initial description in China two-step disease progression with, for the vast majority of the patients, a mild to moderate presentation of the disease during the first week and the first 9 days, followed by, in some of the patients, by a secondary respiratory worsening that arises 9 to 12 days after the first symptoms. So this two-step evolution of the disease raised the question of the implication of the immune response in this secondary worsening. From the first analysis in the Lancet or other publications on the immune response, the fact that the patients had lymphopenia was found in 2/3 of the patients, and the comparison between the severe patients admitted to the intensive care unit, the ICU, compared to the patients in the conventional ward, showed that there was strong modification in some of the cytokines levels, especially cytokines like TNF or IL-6, and also among chemokines. And so even if it's not really still an appropriate probably definition, this worsening of the patient was called the cytokine storm because of the uncontrolled activation of the immune system, and recently some data suggested that in comparison with other acute respiratory distress syndrome or sepsis, probably that the RDS related to COVID-19 is not as an important cytokine storm. So that's why probably that the term of inadapted or exaggerated inflammation is probably more appropriate than cytokine storm. The other stuff is that from the previous studies on the SARS-CoV-1 and the MERS, so the other beta coronavirus, have shown that these viruses were able to evade to the immune system. And you can see here some data doing a summary of what was previously shown and showing that, as you can see, the SARS-CoV-1, but also the MERS, were able to antagonize many different aspects of the immune response. Very recently, also for the SARS-CoV-2, you probably show this study showing that the non-structural proteins, so the NSP proteins from the SARS-CoV-2, were able to antagonize and to disrupt some of the cellular processes involved in the host defense. As you can see, the NSP16 is able to suppress the global messenger RNA splicing. The NSP1, for instance, is able to inhibit the messenger RNA translation and decrease the production of interferon beta, so some of the type 1 interferon. And the NSP8 and NSP9 were able to interfere with the trafficking from the cells to the membrane. And so all of these data suggesting that the SARS-CoV-2 was able to antagonize some of the essential cellular processes leading to potentially in suppressing the host defense. So because of this severe presentation of some patients a few months ago and during the first wave in France of the pandemic, we initiated with colleagues from the Pasteur Institute, from the Imagine Institute, and from the Cochin Hospital, a project that aimed to analyze and to understand what could be the underlying mechanisms associated with a severe disease. So to test the hypothesis that this hyperinflammation that was described at the beginning of the pandemic and the fact that it could be a SARS-CoV-2 driven hyperinflammation leading to the severe disease, we used an integrative approach based on the combination of clinical and biological data, the in-depth phenotypical analysis of the immune cells using mass cytometry. We also analyzed the whole blood transcriptomic analysis based on the NanoString and counter-immunology panel using path-gen RNA tubes, and also some cytokine measurements. So in this initial study, we analyzed 50 patients with COVID-19 with various degree of severity. And we decided to include patients that were not presenting with concomitant comorbidities because we wanted to analyze really the impact of the virus without the confusion factors of concomitant comorbidities like such as diabetes, such as overweight. And so we also included patients with healthy controls without the disease. And what is important to note at this stage is that all the patients were at the same duration of the disease. So all of them had a median duration of 10 days after disease onset, because we know that the immune response against the virus can be really dynamic, and that patients at 5 days of from disease onset 10 days or 15 days is probably not comparable because it's a dynamic response. So some of the patients had mild to moderate disease. So it means they were hospitalized, requiring oxygen, but less than 3 liters per minute. Some of patients were defined as severe patients because they were receiving oxygen at a rate higher than 3 liters per minute. And the critical patients were the patients requiring mechanical ventilations in the intensive care unit. As you can see, when we look at the standard inflammatory parameters, the CRP levels was highly correlated with the severity of the disease. The lactico-dehydrogenase, which is a marker of cellular damage, was also correlated with the severity. And as you can see here, the distribution of the lesions using chest CT scan were showing that more the severe patients were, more the extent of the pulmonary lesions was high. So first, when we analyze the peripheral blood leukocytes phenotyping using mass cytometry, we observed using what we call the Visny representation, which is in fact a way to represent in 2 dimensions the distribution of the cellular populations. We were able to see and to confirm that there was a decrease of the lymphocytes in patients with severe and critical forms, which was already at this time showed, but we were also able to show that there was a decrease in the intensity of the NK cells and the CD3 T cells that were more pronounced for the CD8 T cells. You can see, for instance, here, the NK cells in blue with the density that was going down, especially in the critical patients. And in contrast, there was an increase in the monocytes populations and also the plasmablast population, as you can see here with the increase of the density. And so these phenotypes of decreased NK and CD3 T cells, both CD4 and CD8, was contrasting with an increase in density of the B cells and the monocytes in the severe and the critical patients. So next, we wanted to evaluate what could be the contribution of the inflammatory pathways in the severe patients. And so based on the whole blood transcriptional analysis, we quantified the expression of around 600 immunology-related genes using the NanoString and counter-immunology panel. And as you can see, according to the status, so the infected patients compared to the healthy controlled, and by analyzing the different stage of severity, we were able to see that there was an increase of some of the genes and a decrease of the expression of other genes. So we identify really differentially expressed genes as a function of severity grade. So to represent these differences, we used the principal component analysis that was able to distinguish the patients according to severity. As you can see, the healthy controls were here, the mild to moderate patients were here, and the severe and the critical patients were overlapping in this PCA representation. And what was quite interesting is that, as you can see, the principal component 1, so the PC1 dimension, was showing some only increased genes, expression of genes with severity. And this principal component 1 dimension was mainly driven by inflammatory and innate immune genes. And as you can see here, and in contrast, the PC2 dimension that was higher for the mild to moderate patients and was decreasing for the severe patient, as you can see here, was mainly involving both type 1 and type 2 interferon responses that distinguish patients with mild to moderate disease, severe and critical disease. So we analyzed, based on the data from the NanoString immunology panel, the expression of genes involved in the interferon signaling. And you can see that some of the genes were increasing and some of the other one were decreasing. And so some of the genes that were upregulated in more severe patients were mainly involved in the expression of some receptors for the interferon pathway, but in contrast, some genes that were included in what we call the interferon stimulated genes, so all the genes for which the transcription is induced by interferon, were decreased in critical patients. So next, we used a more specific approach to be really dedicated to what we call the interferon stimulated genes score. That really is a validated tool to define a type 1 interferon signature. And you can see that in comparison with LC control, there was an increase in the mid to moderate patients that decreased in severe and critical patients. And when we analyzed also not the gene expression, but the proteins, we were able to see that the plasma levels of interferon alpha 2 were significantly decreased in critical patients that in mild to moderate patients. And it was also the case when we analyzed another way to evaluate the interferon activity, which is a functional assay evaluating the ability of the serum of the patients to rescue from the lysis some cell lines exposed to a virus. And concomitantly to that, we were able to see that the viral load of the virus in the blood using a digital PCR, which is a really sensitive way to quantify the virus in the blood, was concomitantly and possibly as a consequence, but it's not a proof of this impaired type 1 interferon, an increase of the viral load. And as you can see, using the NanoString quantification for the interferon beta and the interferon alpha 2, there was no real production of expression of these genes. And when we confirmed this data using qPCR among groups, it was exactly the same thing. And the white column is a positive control of patients with a STING mutations showing an hyperactivation of the interferon pathway. So all these data show that patients with severe and critical COVID-19 had an impaired type 1 interferon production and a lower viral response. And maybe you saw this data in Science showing that some of the patients with severe disease can have some mutations involving the interferon pathway or antibodies neutralizing interferon, explaining no detection of the protein in the blood, what could account for something like 15 to 20% of the patients that had the impaired type 1 interferon activity. We next try to dissect the inflammatory mechanisms and the mechanisms underlying this hyperinflammation. And as you can see, when we focused on what was considered to be the cytokines and the chemokines related genes, we observed that there was increased as a function of the severity. And you can see it was the case for the innate immune response system genes, the cytokine and the chemokine signaling genes. And when we analyzed more in details, for instance, the interleukin-6 pathway and the TNF pathway, as you can see, there was a huge increase of the serum levels of the protein, the interleukin-6, in the blood of the patient, which was contrasting with no detection of the transcript in the blood. In contrast, there was an increase of the genes involved in the interleukin-6 induced genes, showing that there was no detectable transcription of IL-6 in the blood, but a huge increase of the protein, and that there was deactivation of in the blood of the IL-6 signaling pathway. We were able to find the same thing for the TNF. As you can see, there was almost a very only moderately upregulated transcription of the TNF in the blood, what was contrasting with an increase of the protein, as you can see here. And there was, like for the IL-6, a huge increase of the TNF pathway in the severe and the critical patients, and especially TRAIL or TNF-SF10, which is really associated and involved in the induction of the cellular death process. And so it was important to see that the discrepancy between RNA in the blood and the protein measurement suggested that probably the cellular sources of these cytokines were not within the blood, but maybe in the injured lungs or the injured endothelial cells. We also explored the expression of the transcriptional factors. And as you can see, again, using the transcription data, that there was a huge increase of the NF-kappa-B pathway that was really upregulated in severe patients. And we know that NF-kappa-B activations can result from the TNF activation and also from the excessive activation by innate immune sensors like the pathogen-associated molecular patterns, the PAMP, like the virus, or the damage-associated molecular patterns that are usually related by necrotic cells and tissue damage. And once again, TRAIL, which is induced by the TNF, is really involved in the tissue necrosis. So to show what the data we had could dissect the immune response in COVID, we were able to see that if you compare the severe patients to the mild to moderate patients, the moderate patients were able to have an efficient type 1 production and type 1 interferon activity, allowing the clearance of the virus and the resolution of the disease. And in contrast, the patient with fever disease had an impaired type 1 interferon activity that could be related, as I told you, by the virus by itself, but also by genetic host factors or the production of antibodies targeting the interferon. And because of that, we could have, as a consequence, the viral persistence activated the NF-kappa-B pathway, either by the PAMs, the pathogen-associated molecular patterns like the virus, or the tissue damage potentially induced by the TNF that could induce what was called the cytokine storm, or probably more now, the inadapted or exaggerated inflammations leading to the severity, the acute respiratory distress syndrome. And so we were able to publish this data in Science a few months ago in July, and to highlight so these different mechanisms of the severe COVID-19. So just because it was really a team collaborations, colleagues from Cochin, from the Pasteur Institutes, from different platforms were involved, and also a lot Professor Alain Fischer, that you may probably know, which was really a very top-notch pediatrician involved in immunology and immune deficiency. And so that's what I wanted to say at the conclusion. Thanks for your attention.
Department of Pharmaceutical Biotechnology at the University of Pest in Hungary, and I think as well Chief Scientific Officer for the company called Humeltis. We have been starting to discuss Technology Access Program project with Judith. I think it was a year and a half ago now, involving the profiling by DSP of breast cancer tissue specimens. So it's going to be a great pleasure for me to be able at least to review the data that have been coming through from that project. Thank you very much, and I leave it up to Judith.
Thank you, and thank you for your attention and the invitation to present this data. And perhaps it is pretty obvious to point this out that my name is not the last name on this project, and it was my full intention to show you that I was asked to participate in this project by the colleagues, the other names you see on the screen. The main reason I will introduce the project as we go along. That first of all, I would like to share the information about the colleagues who asked for my help. The original idea came from Professor Tabar, who is a radiologist, and he initiated the project, and he was the one who, I don't know, dozens of years ago, developed a novel imaging approach for breast cancer diagnosis. And he was the one who established how important mammography is and early screening for women and to reduce the mortality rate in breast cancer. And he currently lives in Sweden. He is originally from Hungary, and he collaborates with people in Boston in hospitals and universities. The other team who work very closely with Professor Tabar is Zsuzsanna Kahán, who is a professor in medical oncology, and she's the one who treats the patients. So she has another set of knowledge. There are also young pathologists, Dr. András Vörös, Dr. Orsolya Oláh, whose immense help was highly appreciated to make this NanoString screening project forward. And also we needed a fully experienced radiologist, who was Katalin Ormandy in this set of selection of samples to be able to show differences, if there were any, in this particular analysis. And because what they saw, they are clinicians, so they saw a different, they saw the whole breast cancer issue from a totally different perspective, and they asked for my help to do something they would never thought of, because of the differences in our view of challenges in the medical field. And because I'm an immunologist as well as a biotechnologist, I could give the kind of molecular view of the whole project, what they were not really thinking of. And the why the breast cancer is so important to investigate. Just a couple of things I would like to mention. That aging of the certain parts of the world is really increasing the demographic pattern in breast cancer, and increases every kind of cancers, of course. And this, if we look at the United States, we find that the highest reported cancer incidence in Florida, Texas, California, and New York. Why? All people move to California and Florida and Texas because it's warm and nice. So they basically, the cancer incidence is highest in those states in the US. If we look at Europe, the Central and Eastern European countries, including Hungary, is the highest in the cancer. Well, why? Hungary is one of the oldest countries in Europe. I mean, the age demographic perspective. And amongst Hungarian women, the younger age group, breast cancer has the highest morbidity and mortality. When we move to Russia, Russian women have the much lower incidence rate than the United States, and it is much more likely that the age differences per 100,000 people is much likely to be much lower in Russia than in the United States. However, the mortality rate is higher. Why it is the case, that's you probably know better than I do, and but it can be the insufficient screening. It is not that it's not available, it is that maybe women don't just go there. And that's what László Tabár is trying to advocate, that really people have to go and see for themselves whether they have breast cancer or not, because that is very important when it comes to treatment. And just a couple of things about the breast cancer for those of you who haven't investigated this area, that basically tumors determined by this particular type of investigation, what László Tabár developed, is mainly where the tumor originates from. And this also not just provides a novel classification system of certain types of tumors, breast cancer tumors, but also it predicts what kind of treatment response these tumors are going to give. And that is a very important one. And these are basically 4 different types of tumors, and I will show you those. And we also need to know that the breast cancers, more importantly, the 2 groups have a significantly different term of outcome. And the more molecular differences for targetable therapies have not been identified. So this is where I came into the picture. Because as I will show you here, and if you are interested, I show you the data or the origin of this data on the corner of the screen. The different types of tumors and subtypes of the breast cancer tumors have, but some of them have very high risk of reoccurring, and very likely that the patient will die. It doesn't really matter what type of treatment is provided. And even in the early stages, that can be the breast removal, the tumor removal is not sufficient, and the people will die anyway. And that is what has to be avoided by the early screening and understanding of what kind of therapy would be useful, or maybe simple markers predicting that the therapy sufficiency in these subtypes of tumors. And also this is what László Tabár has collecting since, as you can see, since 1996. And this is an ongoing collection of data, the different subtypes, morphologically different breast cancer subtypes. He is collecting continuously and investigating the survival rates. And that is how and why they want to know that what treatment would be useful for these particular cases. And here you can see that it is a 26-year cumulative survival of women of this many years. And depending on the morphology of the tumor and the subtype of the tumor, it can be the long-term survival 90% or 54%. And that can be originally determined based on just looking at the mammography picture. And that is a very important issue. This is the 4 types which we eventually selected and investigated. It's not that common, I must say. And so it took us quite some time to collect enough of these tumor subtypes to be able to analyze them in various ways. So here you can see the mammographic pictures of the round mouse, the stellate, or sometimes it is called the star-like subtype of the tumor, and the casting or neoductogenesis, which is frequently HER2 positive, but it's not always. And also the diffuse lobular mesenchymal type, subtype of these 4 tumors, what we investigated. And the overall aim of the shared project was to study molecular heterogeneity in the 4 clinical entities which we set out to investigate, and also the rest of the subtypes of breast cancer would have been the controls for these particular cases. And we also wanted to study whether the immune molecular differences would shed some light on the differential survival rates of these tumors, and whether it is a possible targetable therapeutic approach here, or we have something better therapies to offer to these ladies who have this, or they are unfortunate enough to have this type of tumors. And finally, to identify the presence of circulating biomarkers. This is also part of the project, and this is also my department's responsibility to provide this data, and they are collecting the samples. But now, today, I just would like to briefly introduce you to what finally we got out from this data. And we selected the NanoString's panel, and we had to collect from each type the FFPE fixed samples. We also provided with the hematoxylin and eosin staining, and we got a lot of help from the NanoString team to select the right types and the right sections for the tumor for analysis. And when we look at the selection of the various regions, the regions of interest, so this is where you need a very good pathologist. Without pathologists, basically this kind of projects don't really go anywhere. So the pathologist, András and Orsolya, selected based on the NanoString instructions, we selected 6 regions of interest on each sample, and we also had a control on the sample, which we knew that it was a HER2 positive random breast cancer tumor sample. So based on this initial region of interest selection, we also have been provided by the results, as we have shown the signals to noise ratio. And you can see, because sometimes if you stain a section on a slide, you can stain, or you can see the holes where just the glass is available for you to look at. That can still be a noise provider for this kind of analysis. And basically, if you look at the first tumor sample, it is clear to see that the pan-cytokeratin shows the tumor, which is the green staining, the nuclei are the blue, and the CD45 and CD3 were also stained for that. So those are the immune cells, obviously, and those are not the tumors. So what we wanted to know, that what was on the tumor and not in the surrounding mesenchymal areas, or just not the tumor itself. So with the panel of data provided here, it's huge. So basically the analysis, what I'm going to show you is not the full statistical analysis. It was initially, I couldn't wait for the statisticians to finish all the data. So I had to rush in and find some of the most interesting ones for myself. And obviously there are many control stainings were provided to do the proper analysis, and we would be able to identify specific target molecules of interest. One of what was interesting for us is the control, which you see in the middle, and we knew that it was HER2 positive, and that provided us with a very good feeling, because we saw that the control, which we knew by the pathologists were positive, was positive in the NanoString staining as well. And the tumor was very highly positive. So we went on with more confidence to analyze the rest of the data, and we found that the star-like or stellate-like tumors were BCL2 positive, and the DCI's, the neuroductogenesis type, was also BCL2 positive. So there's only 2 samples were BCL2 positive. That is already in the literature that they require a different type of treatment approach, which was a really high boost for the oncologist, Professor Kahan. So to have a different approach for these examples. And also we found, sorry, this is the other way around, because this is where the star-like, the B7H3 is positive, because I presented on the graphs the other way around. So the orange ones are the tumor samples. And basically we again see here that the B7H3, which is again an immune molecule, immune checkpoint molecule, not very well investigated yet, but potentially is important in the T-cell evasion of the tumor. And again, this is very important potential therapeutic approach for the oncologist team. And so to be able to, because these are individual samples, we were not happy with, obviously, with just that. We took the BCL2 and the B7H3, and we selected 20 samples each from the 4 groups. So basically, we have run the BCL2 staining on these groups, and we saw that the stellate or speculated or star-like was definitely strongly positive. Then we also have seen that the stellate again with a B7H3 was definitely positive here. So we have now the set of immune approach, which we are going to recreate this kind of tumor samples using our technologies developed by my team and in the department to regenerate the kind of tumor composition, and we can try to immunotreat the tumors and or try chemotherapeutic approaches to see how the tumor cells are going to respond with the knowledge in our background now that we can select the B7H3 and the BCL2 2 molecules where the generally accepted treatment approaches would potentially work, but for these particular tumors did not really show any differences in treatment. So we can try and select. And just to finally to say that this NanoString DTS immune panel screening provided us an aim for further directions, which way to go further, and these really, really difficult to treat tumors, what we can try in the future directions. And only finally, I have to say that the cooperation with the NanoString team was very easy, and they were always helpful, so that whatever we didn't understand, or we got stuck somewhere with analyzing the data, which is, I must say, it's an enormous amount of data, which they finally produce from all these stainings. They were very helpful to us, and they also explained everything clearly. So finally, because my main interest is lung cancer, so one of my colleagues now, she's got the funding to go ahead and do something similar in one of our projects. So thank you very much for your attention, and I finish here.
So our next speaker is Aida from NanoString. She earned a PhD in life sciences from Heidelberg University in Germany. And she has a background in tumor microenvironment and immunology. Aida also has experience in flow cytometry, gene expression, and mouse models. And she's now a part of the field application scientist team here at NanoString for the EMEA region. And she's supporting and consulting customers all across Europe, Middle East, and Africa. And she will be doing the software demo for spatial for geomics today. Aida, over to you.
Sorry, can you hear me?
Yes.
All right, thank you for this nice introduction. Oh, let me turn off everything else that I don't need. And apologies about that. So let me share my screen. Hopefully you now can see my presentation.
Yes.
Does it
Yes,
look right?
Perfect.
Okay.
Thank you.
Perfect, thank you. So thanks for the introduction. Today we're going to do this geomics DSP software demo. And before that, I will quickly just go through what Alex already presented you, right? So we know that now the geomics digital spatial profiler can allows us to detect all type of targets. And in this demo, we will see data from protein. We can analyze any type of samples regarding of fresh frozen or FFP tissue, right? And then we can test this data because it's now published in very high impact factor journals. And we can see that it's robust and reliable, not only from data from NanoString, but also from our customer sites. So I'm not going to go again into the process on how the geomics works, but I want to highlight that everything that occurs into the instrument, it is actually controlled in this software that we're going to see today. Okay. So also importantly, we have many times questions about, all right, so if we want to send you these samples because we want to test the technology, but we don't have access yet to an instrument, how is this going to work? Because we want to see the ROI selection, for instance. So this is something we're going to see again and today. And this is really nice because if you have the instrument on the same, let's say, on the same network, you're going to be able to either access directly the instrument that it's placed in this network, or you're going to be given access to the software to be able to select yourself your regions of interest, for instance, or to get access to the data. So how does actually that work? The software in the computer that is, that comes with the instrument has two kind of main parts. One is the geomics control center, which is the one that you will be using for setting up the run and setting up these regions of interest. And the geomics data analysis software part which will allow you to collect, so after collecting the data, to interpret it and to put it into your images. And for this part, this requires actually a very extensive, most probably, talk to understand more deeply how the data analysis is performed. But I'm going to show you a bit on, how intuitive or maybe how user-friendly can that be. So shortly, I just want to highlight that you have several ways of doing segmentations. And for this demo, we're going to see only the geometric and the segmentation one. This is again a summary to show you how this works. So in general, when we have the scanned slides, we have taken an image with immunofluorescence. So as a reminder, we have 4 channels, right? And that one channel has to go for the nuclear staining. So we have 3 more markers that you can choose to visualize your tissue. And this, for example, what we see here in the immunofluorescence staining. And then we have the other reagents, which are actually the proprietary ones from NanoString, as Alex explained, which will be used for acquiring our data. And our data is acquired once we have selected our regions of interest, and then the instrument will apply only the UV light on these areas and will collect the DNA oligos from these areas into different wells that later on we can recapitulate when we put the data in. So what I'm going to try to show you today with our software demo is some data that we've collected in colorectal cancer sample slides. So we, for those who are not maybe that familiar with colon tissue, I would like to start you just shortly introducing like the histology itself, because actually in DSP this becomes a really important topic. So histologically, the normal healthy tissue of the colon looks like this. So these are kind of the islets, and what we can see is that in one of these islets we have the epithelial cells, and this is the cytoplasm, and actually this is the nuclei. This would be the lumen, and then this let's say islets are actually surrounded by very strong stroma. So the tumors, develop from these epithelial cells, and actually they have several stages of differentiation. So you might have neoplastic tissues that are still, having this kind of structure where the cytoplasm of the epithelial cell and the nuclei can be nicely differentiated, but you can also end up with a complete overgrowth of this and only seeing nuclei and stroma randomly. So this is something that can be very nicely observed also in our genomics, right? And this is an image that I will show you today in the software. And what we see here, for example, is these structures, which we will call actually the healthy-like tumor or healthy surrounding tumor. And then here, this is very characteristic of colorectal cancer. So we have a very strong infiltration of lymphocytes and also very nice stroma surrounding it. And this is observed here in the image. And then we have in green, the pancytokeratin differentiated epithelial cells, right, that are, let's say, identify our tumor mass, right? So in red here, we see our CD45 leukocyte compartment. And again, so this is what we would call our healthy colon, and this is our tumor. So there's an image, again, acquired with our genomic CSP instrument. And what I would like to show you is that you can put areas of interest in any part of this tissue, right? And what is the appropriate one? Well, here, the most important thing is that you need to think of which biological question you want to ask, right? So do we want to only interpret if different grades of differentiation of the tumor tissue express different genes on the epithelium? This can be one interesting question. Do we want to, compare this normal healthy tumor versus this tumor tissue? Do do we want to analyze the tumor microenvironment actually on this invasive front versus the tumor differentiated or the healthy like tissue? So with that, I want to just, open up the questions that you can ask with the geomics, and let's go to see how this server works. So because of the demo purposes, I just logged in already. So this is, I hope you now can see my internet or the software. And
yeah,
I hope. Okay, perfect. Thank you for confirming. So then in here, what the first image actually that you will get is this control center. So the first screen that you will get into. And here's where you would start your run, or you will upload your counts. So here what we have is some images that were already acquired by our teammates in Seattle. And now it's loading the scan, so bear with me. I should have not gone back because this takes a bit of time, but should be fast enough. So what we can see is that some slides that were already scanned or that were, readout complete will be the ones from colorectal cancer. So I cannot show you how to select ROIs on these samples because the data was already acquired. But we do have some slides where we can go inside, and just for the demo purposes, let's say we don't care now on the tissue we are seeing. But here, for example, this is the small cell lung cancer tissue. What we see is the whole picture of the tissue that we scanned. And this is how it would look like if you would have your tissue in Seattle, and they would have arranged with you that this is the day that you select your region of interest. And then what you will do, so for example, here you can see that I had already prepared 2 regions of interest randomly. But let's say that we want to, go into this area here. And we are interested in this area because it has very nicely tumor cells that are surrounding the immune compartment. So again, here, apologies about that. So we have a staining of this small cell lung cancer tissue that has been stained with pan-cancer cytokeratin antibody shown in green, and in red we can see that it's the CD45 immune compartment, and in blue we see the nuclei. And if I go deep in, so the resolution will get better, I promise. We can see that in all the time we get this scale bar, which is very nice. So we are now 50 micron zooming in, and you can see that the resolution here is super nice, right? If we go now to these icons here, we have the geometrical arrow, for instance, and we can see, we can draw a new ROI. So this new ROI now will be appearing in here, and we can select it and get into the information. So this is now ROI number 13, because most probably we will have been playing with this, and that's the number it gave. So if you press in the middle, you can move it to your area of interest. If you want to keep your ROIs on with the same diameter, you can now write and say, I want this to be 300 microns. So it will automatically make it. And now, as you can see, because we made this new ROI, we don't have any segmentation. So the segmentation is what will define your areas of illumination, right? And what we might want to do here, for instance, is to tell the instrument that we want to collect the information from our tumor cells in one well, and the information from our immune cells in another well. So for that, we go here, and in the segments, we have to define the parameters in the segment definition part. So if you go on top of the channel name, it will tell you what was in FITC, so DNA, Cy3, it's pan-CK, and Texas Red, it's CD45. So we can select our segments as follows. we have several options for every channel. So here, for example, we have our tumor microenvironment, where we say that we won't care about the nuclei staining. We don't want the signal, so it can be the signal positive as include or negative as exclude. We don't want to have signal that comes from the pan-CK, right, green staining. So we will keep it as exclude, and we want to have the signal only from our Texas red channel, which is the CD45, right? So we will select the positive. For the tumor, for instance, we will ignore again the nuclei staining. We could actually include it if we wish, but in this case, we want to ignore it. We want to include the Cy3 positive staining, and then we want to ignore, for instance, the CD45 one. And then we can press generate segments. And when we do that, it will automatically look based on these parameters that we said, it will generate the segments for collection. So as you can see here, for example, there are some tumor cells that are not taken up, and maybe we want them to be taken up, right? So nicely it excludes, for example, the CD45 positive cells. So as we know that Cy3 is where pan-CK staining is in, we will increase our threshold that you can see that we can play around and see if, sorry, in this case, we want to decrease it, that if it's taken or not taken up these cells. And for that, let's see. So if we put this, let's see if we're having a better taken. Oh, it's not working. So we also have some other advanced parameters. And okay, so let's do this. That I mean, for demo purposes, we're not going to get deep into, but let's see if in this it's now working. All right, we can move forward. So this is just to show you that we can play around, okay? And now we can go to our data analysis. So once we've selected the ROIs, we can go and upload the data. And here, I want to show you some data that we've generated with the colorectal cancer slides. So bear with me, this shouldn't take too long. So otherwise I can start. So luckily, when we upload this data into the Geomics DSP software, it won't allow us to move to proceed to analyze our data without doing data QC. Okay, so the data QC, you will see it now, will actually check for the technical parameters based mainly on the readout. So it will, for encounter, so the protein panels have to be read on an encounter instrument. And in there, you will see that for the QC, we have the encounter basic QC parameters. So if you are familiarized with our encounter panels, you will see that they're the same. So in the QC parameters, we have, for example, the binding density is going to be one of them, and the fields of view that were imaged. So everything related mainly to the readout. Once we have done the data QC, you will get some, let's say, data points that might have flags, and this will not be excluded for later processing, but it's just a way to alert you. Oh, I'm sorry, it's going very slow now. And I just was doing it this morning, so it was working pretty well. Oh, there we go. Thank you for staying here with me. So shortly, let me show. So this is the QC, and as you can see, all the other options are not activated because this is the initial data set. And after you, before you do this QC, you cannot process the data for normalization or scaling. So again, here are the QC parameters, and I'm not going to run it now because I have done it already for the demo purposes. But now once you have done the QC, in this case, for example, I asked that please show me if there are any areas of regions of interest that had less than 100 nuclei and 500 micrometer of area. And because our ROIs were chosen with these parameters, then we have no ones popping up wrong. So for sake of time, let me show you just that how, for example, the data looks like and what can you do here on the visualization. So once you have normalized and you have done your housekeeping normalization and you also have scaled your data for area, you can have always your normalized data on, let's say, on exporting format. So you can see the summary, but you can also from here take this data and bring it to an Excel format, so export it. Then you can do the clustering. And for clustering, for example, we have here the tumor and the tumor microenvironment ROIs that we've selected. And here in the tissue type, we had instead of the tumor and the healthy ROIs. And what we can nicely see here is that, for example, our tumor segments, cluster together very well. The tumor microenvironment segments cluster as well very well. And the tumor ROIs, we have a mix. And then if we want to go and see, for example, only the proof instead of all the proofs, only our myeloid segment proofs, because we're interested in the myeloid compartment, we can also do that. We can group them again. And you can see that it's very intuitive and it's very easy to work with. So for example, if we do a box plot, this is something that we like very much. So you could go and say tumor and tumor microenvironment, select the tissue type in here, for instance, and then you would see how these genes compare in our group of ROIs. Or for example, if instead of doing segments, we do targets, we could see how these targets, for example, the CD14, are expressed in our two groups. And we can see nicely the CD68, for instance, which might be expected as increase in our tumor microenvironment segment. And with that, I think I'd like to finish because I don't want to take you more time. But if there are any questions, I'm happy to answer.